Hello everyone. So in today's session, let's discuss about what are biofertilizers and what are the different types of biofertilizers. So as you all people know, biofertilizers consist of two words if you just see here. So one is bio, the other one is fertilizers. So we know bio means life and fertilizers, yes, they play an important role which enhances the growth of your plants. Now let's check out how are your living organisms play an important role in enhancing the growth of the plants and which are those microorganisms or any species which is involved in enhancing the growth of your biofertilizers. So how do you define a biofertilizer? So they are natural fertilizers that are microbial inoculants of any microorganisms as mentioned here. So it includes your bacteria, algae, fungi, and these microorganisms can be in separately or in combination. So what are the functions of this particular microorganisms when they perform or act as a fertilizer? So they help in biological nitrogen fixation for the benefit of plants. So fixation of nitrogen is a vital step which is involved in the development of a plant. So that process, nitrogen fixation, is taken care by your microorganisms when they are used as fertilizers. And also they help build up the soil microflora and thereby the soil health. So under any conditions like when there is heavy rainfall or drought conditions or when there is flood condition, there are chances the soil loses its fertility. So if soil loses its fertility, then it affects the growth of the plants. So in such cases, when you use biofertilizers, that helps in the maintaining the healthy conditions of the soil, the fertility of the soil. So your biofertilizers not only includes your microorganisms but also include organic fertilizers, for example like manure, your lignite, peat, etc., which can be used as a carrier materials. In later upcoming classes, let's discuss what are carrier materials. So use of biofertilizer is recommended for improving the soil fertility in farming. So these are some of the key features of your biofertilizer, how they play an important role when they are used as a biofertilizer in the development of a plant. So yes, so this slide gives you the overview, the functions of biofertilizers. So as already discussed, yes, it supplies mixture of nutrients, maintenance of symbiotic relationship, maintain microbial constraint in soil, enhanced growth production of crops, suppress the soil-borne pathogenic diseases in crops, and improves your soil health. Now let's move on to the different types of organisms which are involved in the production of biofertilizers. So biofertilizers, as I mentioned, there are different types. So you see nitrogen fixers, that is nitrogen fixing microorganisms. Then you have your azolla, that is your normal fern. Then phosphate solubilizing microbes, vesicular arbesicular mycorrhizae, which is an example of fungal. Then plant growth promoting rhizobacters, sulfur solubilizing microbes. So these are the different types of microorganisms which is used in the production of your biofertilizer. So coming to the examples of nitrogen fixing bacteria. So here examples includes bacteria and blue-green algae. So your bacteria's examples are your rhizobium, azotobacter, bacillus species azospirillum. On the other hand, your blue-green algae, that is your cyanobacteria. Other examples includes Anabaena, Nostoc, Toliphothris, Anobanocyps, etc. And examples of your azolla species are Azolla folliculoides and Azolla rubra. So these are the different examples of microorganisms which is involved in the production of biofertilizer. So in today's class, let's focus on a specific nitrogen fixing bacteria which is rhizobium. So yes, so bacterial biofertilizers. So what are bacterial biofertilizers? How they are involved in the enhancement of the crop production or your plants. So this bacteria consist of live cells. So we know, yes, live cells are present. So in your bacteria, the live cells are used as your biofertilizers, and these bacteria contain unique gene which is called as NIF gene, NIF gene, which makes them capable of fixing nitrogen. So as I already mentioned, bacteria is involved in a mechanism what is called as fixing nitrogen. So that fixation of nitrogen is taken care by the presence of a specific genes in bacteria, and those are called as NIF genes. So this nitrogen fixing bacteria work under two conditions. One is symbiotic relationship and the other one is non-symbiotic. So what is the symbiotic and non-symbiotic? So symbiotic means they develop some formation of structures in any part of the plant such that there is a direct relationship between the plant part and your microorganisms. On the other hand, non-symbiotic, there is no direct relationship. It is as simple as that. So the symbiotic bacteria makes an association with your crop plants. So this symbiotic, where is the association formed? It is in the root of your plant, and there is formation of nodules. So if you take any leguminous plants, for example like fenugreek, etc., if you observe the roots of fenugreek plant, you observe tiny circular structures which is nothing but your nodules, and those nodules consist of your symbiotic bacteria, which is your rhizobium. So that is one of the best example to study the structure of your rhizobium in the nodules, in the form of nodules in the roots. Now coming to non-symbiotic bacteria. So they are non-symbiotic. There is no any direct relationship between the plants and the microorganisms. They are free living bacteria, do not form any association but live freely and fix atmospheric nitrogen. So hope this is clear. So two types of nitrogen fixing bacteria, symbiotic and non-symbiotic. Symbiotic, how are they formed? By the association with crop plants through forming structures called nodules in their roots. And then there are free living bacteria, that is non-symbiotic bacteria. They don't form any association. They are free living and but help in fixing nitrogen. So now let's take an example of your symbiotic bacteria. So as mentioned, an example of your nitrogen fixing symbiotic bacteria is your rhizobium. So yes, so if you see here in this picture, this is one of your leguminous plant and you have your root nodules in the root structure. So you see here smaller circular structures which are formed in the roots. They are nothing but your root nodules. So here if you just microscopically view this nodules, you will be able to observe the rhizobium. So nitrogen fixing bacteria like rhizobium fixes the atmospheric nitrogen into nitrogen compounds. So hope you all got an idea, girls, what is rhizobium, how are they associated, that is how do they maintain that symbiotic relationship with plants. So yes, rhizobium is a soil habitat bacterium. So they colonize the legume roots, fix the atmospheric nitrogen symbiotically. So they are the most efficient biofertilizer as per the quantity of nitrogen fixed constant. So the major role of this root nodules, the bacteria which is present in the root nodules, is that to fix nitrogen. So because of this specific characteristics, they have the wide range of applications when they are applied in the production of biofertilizers. So there are 7 genera and highly specific to form nodules in legumes, and they are referred to as cross inoculation group. So here this group of rhizobium, so we are focusing only on rhizobium bacteria. So they have 7 genera. They are specific to this formation of structures, what is nodules, and this group of 7 genera, they are referred to as cross inoculation group. So hope you are clear, got an idea of how is rhizobium, how is it associated with your leguminous plants. Now let's check out what are the steps involved in the production, commercial production of biofertilizer using your rhizobium bacteria. So you see here, this tabular column gives you the quantity of biological nitrogen fixed by our rhizobium in different crops. So this, when you use a rhizobium as a biofertilizer, it can be applied for a variety of crops. So you see here it is pea, that is pea crop. The rhizobium species from where it has been derived or where the biofertilizer is produced is Rhizobium leguminosarum, and the crops for which this can be applied is for green pea, lentils, and the nitrogen fixation per kg per hectare is about 62 to 132, and so on. There is a list of other rhizobium species which is specific for specific specific crops which when applied it yields maximum, or I mean it fixes maximum amount of nitrogen per hectare. So yes, if you go through it, I think you'll get a clear idea of what is the yield or what is the total quantity of biological nitrogen fixed. So let's move on. Coming to the mass production of bacterial fertilizer. So till now we got an idea what is a biofertilizers, what are the examples of biofertilizers, and we started with a nitrogen fixing bacteria which is Rhizobium. So Rhizobium, it is a symbiotic formation. It resides in a symbiotic relationship with the leguminous plants and fixes the nitrogen. It fixes the nitrogen. So now let's take a deeper look into what are the steps involved. So obviously when we have to commercially produce some products, we have to first need the bacterial starter culture. We have to first confirm that we have a good number of microorganisms, then go on to small scale production, then larger scale production, storage, etc. So in today's class, first let's focus on how to isolate your specific bacteria of interest, which is your Rhizobium. Then identification, that is confirmation that whatever we have isolated is only Rhizobium and there is no any contamination, and check out how can we do cultivation and mass production. So to begin with, isolation. So isolation is very important step, and you see to that there is no any contamination introduced into the culture process. So yes, so these are the steps which explains you the complete steps involved in the isolation process. So the first one is the leguminous plants are carefully uprooted from the soil and washed under running tap water. So you can just uproot your plant sample, and we know yes your nodules are present in the rooter section. So without damaging the plant roots, you're just uprooting it. See to that you're cleaning the soils, remove it completely and washing it with tap water. And then you're going to take only the nodules. We are not going to take the entire roots, but only with the use of a forceps just cut the roots. Which roots you are going to be selecting? You have to select a healthy, unbroken, firm and preferably pink nodules. So if you just check the roots of the fenugreek, there will be tiny nodules which is pinkish in color. So if the nodules are very pink, that indicates it is very healthy. So when you're after uprooting, after cleaning it, make sure you're not damaging the nodules and take only the nodules and further process for what is called as surface sterilization. So once the nodules are selected, clean it with distilled water, that is wash it with distilled water and then processed with surface sterilization. So you people know surface sterilization is done inside a laminar airflow. So please do maintain all the necessary physiological conditions which is required. See to that you do not contaminate your samples. So surface sterilization, yes, the first one is after washing it with distilled water, your nodules are immersed in 0.1% acidified mercury chloride and then leave it for 10 to 20 seconds and wash it with distilled water and then 70% ethyl alcohol twice or thrice so that you remove all the contaminants which are present on the surface of your nodules. So by following all the steps, your nodules are sterilized. So next, what is the step? After washing, so after completing your 70% ethyl alcohol wash, your nodules has to be crushed in small amount of distilled water. So take your nodules into a petri plate, then squash it or just crush it. Gentle tap, they are very soft. Just a gentle tap with the glass straw, it will be crushed and then you are going to culture it on a suitable media. So which is the suitable media for the growth of Rhizobia? It is Emma media. So the suspension is spread on Emma. So what is Emma media? It is yeast extract, mannitol, agar media. So prepare the medias of required quantity, autoclave it, keep it ready and then suspend it into culture flask or onto your petri plates, leave it for incubation. So for here, the suitable temperature which is required for incubation is 25 to 28 degrees Celsius and for 8 to 10 days. So this is the incubation temperature, and you have to leave it for 8 to 10 days. So these are the steps how you are going to isolate your rhizobium bacteria from root nodules. Hope this is clear. So with that we finish isolation. Now let's move on to the so this is the schematic diagram how we are going to, what are the steps involved in your isolation. So you see here, root nodules are taken, take only the nodules from there, wash it, surface sterilize it and crush the nodules, and then you, if preferred, you can go for serial dilutions, and from specific serial dilutions like 10 power 3, minus 3, 10 power minus 4, 7, 9. Similarly, you can plate it on your petri plates on your MR media. So after that, you are going to incubate at 25 to 28 degrees Celsius, or you can also put it onto agar slants for about 8 to 10 days. So after 8 to 10 days, you will be able to visualize the colonies, which indicates the formation or presence of rhizobium. Now, how are you going to identify or confirm whatever colonies are formed on your petri plate is rhizobium? So we don't know whether those colonies formed on your petri plate is rhizobium or not. So for that, we are going to study how to make or how to come to a conclusion and say yes, those colonies are only rhizobium. Let's check out how to do that. So yes, identification. So see here, this is a petri plate which consists of your MR media, and you see there are small white colored colonies formed. So this, yes, according to us it is rhizobium, but how can we convert? How can we confirm those are your rhizobium? So for that, we have to know the characteristic features of your rhizobium. So here, the rhizobial cells possess characteristic refractive granules of beta hydroxybutyrate within the cells. Okay, so because of this compound, what is that? It is beta hydroxybutyrate within the cells. So because of this, there is characteristic formation of rhizobium when grown on an MR media. So what are those? So they appear as smooth, elevated and glistening white colonies, that is glistening, slightly shining, and they are very smooth surface. These are the characteristic structures of your rhizobium, and that can be further confirmed by your gram staining techniques. So here for gram staining, we are going to perform it using carbonyl fusion stain. So when you stain your colonies with your carbonyl fusion stain, what is it? What are the how do you identify it as under microscope? Is that it appears as gram-negative rods, it is motile, not spore-forming and pleomorphic bacterioids. So it is a gram-negative rods, that is you are going to observe purple-colored rods when observed under microscope. So that's how we are going to confirm, okay, these are my rhizobium without any contamination. So this is one of the method. How can we confirm the colonies which are formed on the MR media are a rhizobium? So the next one is confirmatory test. Yes, even after the staining procedure, we are not able to 100% say they are purified cultures or starter culture of your rhizobium. Then you can go for a confirmatory test. So what is this confirmatory test? It is nothing but the growth on Congo red yeast extract mannitol agar media. So the media remains the same, that is YEMA media, but you are going to add a indicator. What is that indicator here? It is Congo red. So here when you culture your rhizobium on Congo red yeast extract mannitol agar, they form white, translucent, glistening, elevated and comparatively small colonies on the media. So can you make out the difference, girls? Here the colonies are very big when compared to the colonies which is formed on your Congo red media. So by this you can come to a conclusion, okay, these are my rhizobium colonies. So moreover, the one most indication is that our rhizobium do not take up the color of your Congo red dye. So it is just on a red background. We are going to visualize white colonies on the media. So those colonies which readily take up the Congo red stain are not rhizobia. So if you observe any colonies which is color rather than white colonies, it says it is an indication that those colonies are not rhizobium but other bacteria, which is preferably agrobacterium. So these are the two methods how you can identify and confirm whether the colonies which are found on the Emma media is rhizobium or not, and see to that how you get pure cultures of rhizobium. So hope this is clear. So we discussed about isolation, how we are going to isolate under laminar airflow. Then for after the growth incubation or after the growth of colonies on the petri plates, how are you going to confirm or identify only those colonies are of rhizobium and are not any other contaminated bacteria. So next, let's move on to cultivation and mass production. So yes, now we have the rhizobium pure cultures. We have to cultivate and see to that how it can be produced, mass produced for the production of biofertilizer. So coming to the cultivation process. So cultivation is somewhere where you are going to obtain your starter culture, a good starter culture of rhizobium. So what should be done here? So prepare appropriate media for specific to that bacterial inoculant and sterilize. Then the media is inoculated with efficient bacterial strains. So here the media will be Emma media. Prepare it, sterilize it, autoclave, sterilize, cool it and keep it ready. And then once your colonies are ready, rhizobium, go in inside the laminar airflow, inoculate your media with your rhizobium under aseptic conditions. So after that we have to do incubation. So here incubation is done in shaker incubator. So when you place your conical flask in a shaker incubator, it makes sure that there is more space given and there is more aeration such that it gives you good result of growth of rhizobium. So incubate the flask under room temperature in rotary shaker at 200 rpm for 5 to 7 days. So what are the conditions in shaker incubator? The speed is about 200 rpm and the incubation period is about 1 week or 7 days. So now after 1 week you will be observing the flask for growth of culture. Check out what is the population, that is total number of viable cells present in your culture media, and that total number of viable cells which is present is nothing but your starter culture. So now using this starter culture we can go it for mass production. So how it can be done? So we know in the developmental phase, in the growth phases, we have different phases like lag phase, log phase, stationary phase and the decline phase. So which is the best suitable phase which has to be selected for the mass cultivation is that. So using the starter culture at log phase. So when during this development you have to, on regular intervals of time, you have to check on which phase your rhizobium is growing. So at the log phase you have to block the growth and see to that they are inoculated into the larger flask for mass production. So the starter culture you are going to take at the log phase. So these are the steps followed for the cultivation process. Hope this is clear, girls. Now let's check out what are the steps involved in mass production. So yes, mass production. So as I mentioned here, we are going to take the log phase from the starter culture and inoculate into larger flask. So for mass production, your conical flask will not be good enough to grow your rhizobium at a larger scale. So here we are going to make use of larger instruments, what is called as fermenters. So yes, so in the fermenter your media, that is your Emma broth media, is prepared in large quantities, sterilized and cooled. So at the 5th day or 7th day, prepare your required amount of media, add it into the fermenter, see it is sterilized and cooled. And then the media which is present inside the fermenter you are going to inoculate with your rhizobium. So at what phase? It is at this log phase of your starter culture. And then the cells are grown in the fermenter by providing aeration. So obviously in the fermenter there is complete stirring of the media and the microorganisms. So again it is similar to your shaker incubator. So that provides your good aeration and gives your continuous stirring. And then So after it is not advisable to store your broth, that is your media, after fermentation. So after a period of 2 to 3 days, your media completely will be fermentation would have been completed. So once after it is done, see to that you immediately use it and proceed with the cultures. If not, it is not advisable to go for longer than 24 hours. So even though you cryopreserve your cells, you might lose your number of viable cells there. So it is not advisable to delay the process. So once after this, the broth is checked for the population of contamination, if any, and there shouldn't be any contamination including your fungal or any other bacterial contamination when you dilute your broth for about 10⁻⁻⁶ dilution level. So once you check your contamination level, then your cells can be harvested with a population load of 10⁻⁹ cells per ml after incubation period. So this is how you harvest your cells under a large scale production. So media is taken in the fermenter. You either start a culture in the log phase, leave it for continuous stirring aeration, check for the growth, total number of viable cells, check for contamination. If there is any contamination, see how it can be avoided, and finally after the removal of contamination, you are going to harvest it. So how much ml of cells you are going to harvest? It is for the population load of 10⁻⁹ cells per ml. So by this we complete mass production steps. So if you look here, this is the schematic representation, that is whatever I have explained, that is your cultivation and mass production is explained here. So this step is your cultivation process. So you see your mother culture is your starter culture on your MR slant and your broth, then broth in smaller flask, maybe 250 ml, then 500 ml, then you keep it in shaker incubator, check for the broth quality checking and proceed. On the other hand, for larger scale, from this step you are going to use the starter culture, that is log phase, and supply it in the fermenter. So after this you check the broth for the total number of viable cells, harvest it, and then before harvesting, that is, you are going to blend it with a carrier materials. Yes, we'll discuss what is a carrier materials. So there will be the starter culture which has been, the cells which has been harvested will be blended with the sterilized carrier peat or lignite. It will be packed in polythene bags, and storage will be about 25 degrees Celsius, curing in control room temperatures. Then you check the quality of the biofertilizer, and completely it can be stored for 4 degrees Celsius in low temperature rooms, and then it can be made available commercially and it can be dispatched to the farmers. So this is a schematic representation which explains you how the process of the production of biofertilizers happens. So hope this is clear, girls. So in next class, let's study about what is the carrier materials and so on. So with that, I complete this topic. Go through the materials and get back if any doubts. So in today's class we discussed about what is a biofertilizer, examples, types, then bacterial biofertilizer which is a rhizobium, and how the steps involved, and then we got to know how the isolation is done, then identification and your mass production. So with this we come to an end of this class. Meet you all in the next class. Take care.